today we are going to discuss about structure and functions of immunoglobulins. So under this we have to study about various type of immunoglobulins, then structure of immunoglobulins and functions of immunoglobulins. So what are immunoglobulins? Immunoglobulins are antibodies which is secreted by the B lymphocytes which is during the course of immune response. And they are a specialized group of proteins which is mostly associated with the gamma globulin fraction. We know that our plasma consists of three types of protein: albumin, globulin and fibrinogen. And this globulins are of alpha globulins is there, gamma globulins is there, and beta globulins is there. So in that, this immunoglobulin is a specialized group of protein which is mostly associated with the gamma globulin fraction, and it is abbreviated as IgG. Then when we are discussing about the structure of immunoglobulins, you can see that this is the structure of immunoglobulin. This immunoglobulin consists of two heavy chains and two light chains. In this picture, you can see the heavy chains and two light chains. And these heavy chains and the light chains are linked together by disulfide bridges. You can see there, they can be linked together by disulfide bridges. And so we are representing this immunoglobulin is we can say immunoglobulin as a Y-shaped tetramer, that is and it can be represented as H2L2. Okay, then why it's called as heavy chain? Because heavy chain approximately consists of 450 amino acids, whereas light chain consists of only 212 amino acids. Then each heavy chain or light chain has two regions, or we can say it as domains, namely constant and variable region. You can see here, when we are looking at the light chain, you can see one variable region and one constant region. When you are looking at the heavy chain, you can see one variable region and three constant regions are there. That is why it is said that in the heavy chain, 1/4 of the amino terminal region is variable, while the remaining 3/4 is constant. Okay, so that is about the structure of the immunoglobulin. And in the, you can the major thing is in that region you can see there is an antigen binding site, and that in that antigen binding site of the immunoglobulin, the antigen will be binding to the immunoglobulin structure. Okay, or we can say it as antigen binding site or antigenic determinants, like that. Okay, then depending on the heavy chain makeup, immunoglobulins are classified into 5 major classes. They are IgG, IgA, IgM, IgE and IgD. And we can remember this immunoglobulin type using a mnemonic called GAMED. In that, G representing IgG, A representing IgA, M representing IgM, E representing IgE and D representing IgD. Okay. Then this is about the structure of the immunoglobulin. In this structure, you can see that IgG, IgD and IgE are monomers. But when you are looking at IgA and IgM, you can see that IgA is a dimer, that is two monomers is joined together by a J chain. Okay, and IgM is a pentamer, and here they consist of 5 monomer unit, and in this 2 monomer units are linked together by a J chain. Okay, so that is about IgM. Then when we are looking at the functions of each immunoglobulin, IgG is the most abundant immunoglobulin and is the major antibody of the secondary immune response. Secondary immune response means we know that when an antigen is entering our body start to secrete antibody against that. So that is called as primary immune response. When the same antigen is again encountering our body will start to secrete immunoglobulins. The memory cells of our body will be getting activated, and that will be remember which antigen has encountered us, so they will start to secrete antibody against that particular. So during that secondary immune response, the major antibody which is secreted is IgG. Okay, and this is the only immunoglobulin which can cross the placenta. So that is why this is how the mother's immunity is transferred to the developing fetus. Okay, then it is also triggers the foreign cell destruction which is mediated by complement system. Complement system is a specialized type of proteins which is mainly associated with the immune response, and the main functions of this complement system is phagocytosis. How they function is mainly by phagocytosis. Okay. Then IgA. IgA is mainly present in body secretions such as saliva, tears, sweat, milk and the wall of the intestine. And IgA is the antibody which is present in the colostrum, and IgA is preventing the foreign substances from entering the body cells. So that is about IgA. Then IgM. IgM, we know that IgM is the largest antibody because it's a pentamer. So it's a pentamer, so it is having 5 antigenic determinant site. Then it is the largest antibody, and it is the first antibodies which is produced in response to an antigen. That is why we can say that IgM are the predominant class of antibodies which is secreted during the primary response. Then the natural antibodies are IgM in nature. Okay, then IgE. IgEs are the antibodies which mediate allergy, and they have the property to fix on the mast cells and basophils and causes the release of the histamine. The substance is called as histamine. Then IgD. IgD are the immunoglobulins which is present on the surface of the beta cells, and it is present in very low concentration in circulation, and they function as B cell receptors. Okay, so that is about the functions of immunoglobulins. And in this slide, each we have represented the functions of each immunoglobulin in one slide in the same thing which we have discussed in the previous slide. That is, IgM is the antibody which is produced in response to, it is the antibody which is produced during the primary response. Then IgG is the principal antibody during secondary response, and it is the antibody which can cross the placenta and crosses the placenta. So this is how the mother's immunity is transferred to the fetus. Then IgA is the antibody which is present in the colostrum of the milk. Then IgD is the antibody which is mainly IgD's function is not to be determined, and but it is the main antibody which is found towards certain antigens. Then IgE is the antibody which mediate allergic, or it is the antibody which is secreted mainly during allergic responses. So that is about immunoglobulins. Thank you.